All right, I'm going to go ahead and get started because I got a lot. Hello everyone. Thank you very much for coming, and thank you very much to the organizers of this, putting all this together. First, I figured I'd introduce myself. My name is Andrew McCall. I'm the director of the Optical Imaging and Analysis Facility, which is part of the University of Buffalo School of Dental Medicine. I'm housed in the Biomedical Research Building right here. My facility houses a pretty wide variety of instruments, not only microscopes, but I also have flow cytometry, cell sorting, and micro CT. So I have a fairly diverse set of tools at my disposal. Today, however, I'm going to be talking, of course, about my recent work on developing a new Fiji spatial correlation plugin that is more compatible with super resolution data. Before I start discussing my plugin, I'm going to give you a brief overview of colocalization techniques. Especially common ones, concentrating a bit more on problems with Pearson's correlation coefficient and similar pixel-wise methods. I'm then going to go over image cross-correlation spectroscopy, and then my plugin colocalization by cross-correlation, which is long, so I'm going to call it triple C. Starting with how it works, some of the newer advanced features, and then we'll get into the tutorial. After that, if there's still time, I'm going to go over how you report some of the results that come from my plugin, how to do stats on it, and any troubleshooting that you may run into with it. So colocalization is currently used as a rather generic term that can be divided into a lot of different types of analyses. And there are a lot of ways to divide it up, and this is the one I tend to use and like. Over here on the left, you can see we have object-based colocalization. This is where you segment your image into objects and background, and then you can take the intersection of your two images compared to the union of those two images or compared to one of the images individually to basically look at how much colocalization in terms of area there is. Another thing that you can do if they don't overlap is you can do a nearest neighbor analysis. With these particular images, with this particular method, after you segment your images, the fluorescence intensity no longer matters. So we're only using that intensity up to segmentation. And then once we get our objects, at that point, we move past it. In contrast, the intensity-based colocalization methods use intensity as part of the algorithm. The most common of these and well-known of these are the pixel-wise methods, such as Pearson's correlation coefficient. Pixel-wise, what that means is that we're just comparing each pixel of one channel to the corresponding pixel of the other. And in this case, what we do, especially with Pearson's correlation, is we take any of those pixels and then we remap the intensity in both channels to a scatter plot of one channel versus the other. And then we use that to look at the correlation between those two channels. Yeah, and this gives us the Pearson's correlation coefficient. Effectively, what we're doing when we do this is quantifying the overlap of those two, but using the intensity to do that. We also have cross-correlation functions. The most common and well-known of these is the fluorescent cross-correlation spectroscopy. What you do with FCCS is you excite a single focal volume, usually with two lasers, and you record into two channels simultaneously, in this case, red and green. And so if your red and green signal go up and down at the same time as you record in this channel, then you have correlation, you have colocalization of those two molecules. They are moving and traveling together. So we're measuring the signal correlation over time. Another cross-correlation algorithm is image cross-correlation spectroscopy. In this, what we do instead is we cross-correlate across the image. So we're moving, cross-correlating over space and measuring things as a function of distance. This is where triple C resides. So you'll be hearing a lot more about this in just a few minutes. And then there are a couple special techniques. So single molecule localization microscopy has some colocalization techniques that are very specific to how they work. And then there's also FRET, which gets down to very, very close analyses in terms of spatial correlation. If your goal, I'm not going to be talking more about these, I'm sorry. If you're interested in these, you can feel free to look them up on your own or ask me about it afterwards, but these are rather specialized techniques. If your goal in colocalization is to try and provide evidence that the two proteins you're looking at are binding, the best ones here are FCCS and FRET, and my plugin absolutely does not change that. The thing about this is FCCS and FRET are both rather difficult to do. They're fairly complicated. So a lot of people, they tend to move from these, instead of doing the more complicated ones, they go to the pixel-wise techniques because they're relatively straightforward and simple. My goal in making CCC is to actually try to get people to move away from these pixel-wise techniques because there are a number of problems with them and go to image cross-correlation spectroscopy. Or at the very least to support their pixel-wise results with image cross-correlation spectroscopy. Before talking about some of the problems with pixel-wise techniques and why I think we should move away from them, I'd like to discuss what we're actually measuring when we're using these techniques. At the most basic level, these methods quantify the overlap of two fluorescent labels in an image, right? It's a little bit more complicated than that, but that's basically what they do. If the signals do not overlap, you do not have correlation. If they do overlap, you're going to have at least a little bit of positive correlation. In reality, we don't actually care about the fluorescent signal. What we care about is the two proteins that our fluorophores are labeling, or organelles, or whatever it is that they happen to be labeling. It could just be molecules. We know that these two proteins cannot overlap at a molecular level, right? If we had infinite resolution, we would not get a zero correlation distance, right? So our true spatial correlation cannot be zero. However, when we're using these pixel-wise techniques, they are only measuring the overlap, which effectively means that they are telling us that that correlation is centered at zero. Since we know the true correlation cannot be zero, there has to be some amount of error, a range across which our correlation exists or below which our correlation exists. This scatter plot does not tell us that range. None of the pixel-wise techniques, Pearson's, Manders, Lee's, or Costa's, significance test, nothing like that tells us what that range is. A lot of people will argue that the range of that correlation is defined by the resolution of the image. And in some cases that might be true. However, you can see in this image here that we have aggregates, protein aggregates. So that is not true with this particular image. When we have aggregates, we're no longer limited by our range, but by the size of these aggregates. Additionally, even if you are limited by the resolution of your microscope, often people do not report on the measured resolution of their microscope. Quite frequently, you will just get the numerical aperture of the objective that was used. If you've done that, sometimes just the magnification. And usually the theoretical resolution limit that you can calculate from the numerical aperture does not match with the actual resolution that people are getting in the microscope. So all these pixel-wise methods are telling you is that there is some sort of correlation at a distance kind of near zero, and that's about all we get. Also, because these pixel-wise methods require overlap, they are not able to measure spatial correlation in images if there is a gap in the fluorescent signals. So if we look at this image where we have nearly perfect spatial correlation, this will give you a Pearson's correlation coefficient of 0. This may not have been an issue in the past when resolution limits were lower, but with the new super resolution techniques, particularly expansion microscopy, we are at the point where we can achieve EM levels of resolution with a light microscope. And these pixel-wise colocalization algorithms are simply not going to continue working. Even putting aside this extreme though, this all points to a more fundamental problem with these techniques, which is that as you make efforts to improve your imaging, as your resolution goes down and down, eventually the overlap will go down and your coefficients will therefore go down. So as you improve your imaging techniques, your results will get worse. Inversely, if you take low resolution or poorly imaged samples, like this one, you are actually able to more easily get a positive correlation result with this, even though underlying that there is no real spatial correlation between those two labeled structures. What this means is that we can easily get a false positive with a low resolution image with no underlying correlation, and a false negative when we have a high resolution image and perfect correlation. So how do we fix this problem? The answer, in my opinion, is we go with a different approach entirely. Those same images can be analyzed using something called image cross-correlation spectroscopy. With image cross-correlation, what we do is we sweep one image across the other, and we measure the correlation at each translation, and then we map that onto a cross-correlation result image. When we do this, we'll be able to see peaks of correlation in that cross-correlation result. So if we take that image from before, we had nearly perfect spatial correlation. Now we can see that ring of correlation around the middle of the image there, and that is our spatial correlation. in this image, at the very center of it is where we have zero translation. And this is where every single pixel-wise technique resides, is in that one single point. With image cross-correlation spectroscopy, we can take the radial profile of this image, starting at that center point, moving out, and actually get a cross-correlogram of our correlation in order to calculate where our correlation lies. In this case, we get these two signals, the blue and the magenta here, correlate at 2.32 microns. We also get a standard deviation as the error of our measurement from our resolution limit. So our resolution limit is actually incorporated into our results. With the same system, if we take this image from before where we had low resolution data and we cross-correlate it, what we get is this very broad standard deviation. This is the type of result that you get when you don't have enough resolution to answer the question that you are asking. All of this here is the generic process for image cross-correlation spectroscopy, which is the foundation of CCC. But I was not the first to develop an algorithm using it. There have been previous MATLAB scripts that use image cross-correlation in this exact same manner to study spatial correlations as well. However, CCC does have some advantages over those MATLAB implementations. The first being that it simply does not use MATLAB because teaching MATLAB to biologists is just not a fun task. So I tried to keep my plugin in Fiji. It's entirely built in Fiji, and I tried to keep it as user-friendly as possible, at least in its execution. It was a little complicated to analyze, but we'll get there later. There are a few options, but there's nothing that requires any knowledge of optical physics or linear algebra, nothing like that. You basically give it some images, a mask, and you tell it what you want to generate and where you want to save that data. Additionally, my plugin is compatible with anisometric 3D images, which means your X, Y, and Z do not have to be scaled the same, instead of just 2D images like its predecessors were. I also figured out a way to actually remove the low spatial frequency component. I'll be discussing a little bit more about what this is and why you need to do it. But for now, if we look at this graph, you can see the blue here is the original cross-correlation that we did without removing the low spatial frequency component. And by the way, this comes from different data than what you're seeing here. We'll see this data in a minute. When we remove the low spatial frequency component, we get this green line, and you can see just a slight difference in how steep this line is here. So it goes down a little bit more, and we kind of remove this background slope to a degree. It's not perfect, but it definitely has changed. This improves the precision of our results quite a bit, and it'll be showing a demonstration of how this works and why we're doing this in just a moment. The last thing is that when we use CCC, we can actually back calculate to get what data contributed to the results, to that Gaussian curve. So we can take the Gaussian curve and use it to basically isolate only the data that's within our correlation range. So if you see in this image, we have these few points that don't have a corresponding pair, and in our contribution images, as I call them, they are missing. The reason I call them contribution images is because if you have pairs that actually line up, those will be brighter than the ones that don't. So you can see in here, we have some matching pairs in terms of their angle, and those are brighter because they contributed more in that regard. And that comes from the cross-correlation result image. So to use CCC, there are a few pre-processing steps that need to be done if you want to get good results. The first thing we need to do is we need to prepare a segmented mask of where we want to analyze for spatial correlation. This is really important in removing that low spatial frequency data. So if we are looking at the cross-correlation between the endoplasmic reticulum and mitochondria, like in this image, we're basically looking within the cytoplasm. So the mask that we're going to use needs to be a mask of the cytoplasm. That's generally how it works. You are not creating a mask of your data. You're trying to create a mask of where your data lies, what it's contained within. That's the general idea. Beyond that, you're not going to need any more masks. You're also probably going to want to deconvolve your data. If you're not familiar with deconvolution, I sadly don't have time to discuss it in detail, but basically it's a form of mathematical deblurring. It's relatively accurate and definitely a good idea. You will get better results when you deconvolve your data. The next thing we want to do is we actually want to try and get the background to 0. And the way we want to do that is generally, if you do have positive background, you want to first change your image to a signed numerical format. In Fiji, that is by going to image type 32-bit so that we can have negative numbers. And then you want to subtract the background from that. any background, even when it's mostly zero, but you have a few positives, will contribute to a gap between your original cross correlation and the subtracted cross correlation, which affects something called the confidence, which we'll be discussing a little bit more later on. So you'll want to subtract a background, and we'll be doing that in the tutorial for one of the images. And then the last thing you do, and I do recommend doing this right before you put it into the plugin, is you want to verify the scaling metadata. The image scaling has to be accurate in order to get this cross-correlogram distance to be accurate. So once you've done all that, you take your images, you take your mask, you put it into CCC, and you will get out, optionally, the cross-correlation result images. You don't have to get these if you don't want. You get the cross-correlogram, just like this, with the distance and the cross-correlation. You will get your metrics for your Gaussian curve fit, as well as your confidence, and the R-squared, the goodness of fit of your Gaussian curve to your data. And then again, optionally, you can get the contribution images. These do not have to be generated. Once you have all that, you will have your results. Sorry, messing up here. One of the most important pre-processing steps here is the mask. So I want to quickly discuss this using this rather simple example. If we were to analyze an image of just random points, and we had basically a process to randomize the points and analyze it over and over again, you would eventually find that there's no correlation, or at least no consistent correlation over iterations, right? However, if we take and do the same process, but instead we add a black background around that image before we analyze it, and we're analyzing the whole region, you will find that there is a spatial correlation over distance. Because we have these confined to a region, they are correlated around zero with this massive standard deviation simply by being confined. And this is analogous to what you have in the cell. Your proteins are confined to the cytoplasm or to the nucleus, whatever container they're going to be in. And you have to basically remove that spatial correlation contribution from your data. I'm not really going to go over how that works, but just know that it is really critical, and I want to demonstrate this with some real data. So if we take this image again and compare our result with the mask and without, with the mask, you can see that we have a mean distance between our endoplasmic reticulum and mitochondria of 590 nanometers and a standard deviation of 570 nanometers. This makes sense. They're not very consistent in terms of the space between them. So we would expect a pretty large standard deviation. Additionally, your confidence is only 0.22, which is pretty good. But you will see in a second, compared to the mask, without the mask, sorry, we have a very similar mean, a little bit longer, but not too different. However, our standard deviation is now huge. We have 7.76 micron standard deviation. And interestingly, our confidence went up. And this is the type of result you tend to see if you don't use a mask, right? And what this is saying, what CCC is saying, is that it's very, very confident that these two particles correlate within this range, which of course makes sense because these two particles are confined to each other within this range because they are confined by the cytoplasm. So you have to use a mask in order to basically get rid of that low spatial frequency contribution in order to get what you're actually looking for. On the subject of confidence, I wanted to talk about it really quickly as it seems to be the most misunderstood metric, probably because I made it up. Confidence is not a measure of correlation. It is not comparable to the Pearson's correlation coefficient, and it does not represent the fraction of the correlating signal. In order to get it, all we do is we take the area under the curve of the Gaussian fit and we divide it by the area under the curve of the original cross correlation within the range of the mean plus or minus 3 standard deviations. And what it's for is it's a metric useful mostly for troubleshooting or filtering results. I still feel it should be reported, but you can definitely put it in supplemental. This is not something that you should really be analyzing. And what it's trying to do is basically trying to ask the question of, do we have enough resolution for the given fluorophore abundance within the mask region? This is a similar idea to the Costa's significance test, but with Costa's, I do find you often get a value of 1, which basically just means everything's fine. With confidence, you definitely get a lot more variation in your data. I will add this can be affected by the fraction of correlation and the number of correlating particles, but also so many other factors that you don't want to read into it in that way. If you're looking at the if you want to look at basically how many particles are correlating with each other, you want to use Gaussian height. And there are just a few things that you need to check before using the Gaussian height, such as you have similar levels of fluorophores in both your conditions, your control and your experimental. If one of them has a lot more, say, green, that's going to affect the Gaussian height, even though it may not be affecting the number of particles or the fraction of correlation. Additionally, when you're looking at the confidence, a confidence greater than about 0.2 is actually really good, and 0.1 is okay. If you get lower than that, you generally want to start looking at how to improve your resolution or improve your images, which can be as simple as just deconvolving your data. Okay, before we get to the tutorial, I wanted to mention a few of the more advanced features of CCC. First is that this can actually accept time-lapse data directly. We're going to be doing this in the tutorial. For those of you who may be familiar, this is the mitosis image from Fiji. So we'll be using the sample image in order to do this exact same thing. So we'll be going over that. And then a brand new feature that I released just a little over a week ago is multi-term Gaussian fitting. So previously, when you were using CCC, if you had a data like this, where we have this really long, broad correlation, and then this kind of narrow one on top of it, CCC would fit to that long, broad one, right? And there was no way to kind of figure out what this value was. Now you can actually fit two Gaussians, and it'll fit a sum of Gaussians curve fit to that data, so you can actually get what this is. I should note that the confidence values are calculated per Gaussian, so this will have a separate confidence, and it's going to be pretty low just because it is so small here. As a practical application of that multi-Gaussian fitting, we're going to look at this project shown here of a transfected viral DNA replication system from Drs. Rama De Rao and Thomas Melendi at the University of Buffalo. Here we are looking at the recruitment of cellular DNA polymerase, which is in magenta, to intranuclear viral DNA replication centers by a viral replication protein, which is shown in green. Okay. With this data, one of the interesting things that we can do with CCC is to quantify the polymerase aggregation in infected cells using autocorrelation. So if we cross-correlate the green and the magenta signal, we get this curve, pretty standard curve, and it definitely shows that our signals are correlating very strongly at 0. However, we can use autocorrelation with just the magenta channel. And what we find is that we can actually separate out this kind of broad curve from a narrower curve. And that broad curve represents the homogeneous component of that DNA polymerase within the cell. And that narrow is these aggregates. What this means is that we can use the formula for the area under the curve of a Gaussian curve and actually look at basically how much data, how much cross-correlation do we have just in that narrow band compared to the total within both bands in order to calculate the aggregation within that cell. So in our transfected cells, we have about 30% aggregation. And I know we can't really see the homogeneous component here in this image. I had to make it more transparent in order to actually be able to make this animation work, but it is there. But we have about 30% aggregation in the transfected cells. And in the control cells, we do see these little puncta still. It's only at about 11.5% in that narrow band there. So we definitely see a difference, which was definitely an interesting result in application. And with that, I will take any questions and stop for the tutorial. Sorry if people have been asking questions. I can't see the chat, how I was doing things. But okay, what represents each individual blue lines in the cross-correlogram? Right, yes. So those individual blue lines are actually the z-steps. So if I go really quickly back to that, right here, these blue lines and the green lines, these are the z-steps. So when we translate the image up or down one, we get a pretty significant reduction in this case in the cross-correlation. And that's actually because this is a cell that's being grown flat on a cover slip. So it has a lot of sample anisotropy. And so translations in Z have a pretty big impact. You won't see this with all data. I'm sorry, I'm not sharing my screen. Well, that was a mistake. Let me go ahead and share my screen here. Hold on 1 second. My PowerPoint is misbehaving. There we go. Okay. So yeah, these right here are Z steps, right? And so the sample anisotropy means that there's a pretty big step down when you were looking at the cross correlation. So I did forget to mention, for those who have joined kind of after the start there, you can find a link to the tutorial data in the meeting information. And if people want, I can actually post it into the chat too. This is a rather small file, so it should not take particularly long to download. Access the chat right here. So there is the link if you need it. And we will go ahead and get started here. Oh, looks like there's one more question here. Can you evaluate exclusion with this approach? Yes, you can, kind of. So if you're talking about very generic exclusion where you just have kind of an aggregate of protein in one area and then more homogeneous everywhere else, the way you would want to do that is you would actually want to invert the more homogeneous channel so that you were looking at where it is not, and then evaluate the spatial correlation with that. And you can basically look at the inverse of that relationship. At least I believe that would be the best way to do it. It's not exactly a direct analysis, but that would be one way to do it. So once you get the data, we're going to start here with a couple really basic examples. So the first one, and you don't have all this, you don't have the composite, but I just want to be able to show you what this looks like. This comes from Andrew Rowe, one of his projects. He's in the lab of, I'll try and pronounce this correctly, Trina Ni Kundela. I think I butchered that, but I made an effort. And this is actually storm data where we're looking at mitochondrial proteins. So you can actually see there is a pretty strong correlation here. And if we take these two images, you go to, I'm sorry, let me back up. Make sure everyone has installed CCC. I'm sure you all have, but if you go to help, update, manage update sites, search for colocalization. It's right there, colocalization by cross correlation. Once you have it, if you go to analyze colocalization, you will find it in here, and we're going to use CCC. There are a couple other options. If people want at the end, I can talk more about those. This is kind of the main one that you'll be using here. When you open this, you get a selection for your two images. So make sure that you have the two different images selected. We want green and red, not green and green. And then this data, we don't have a mask. I've cropped the data to be just within the cell. So you will not need a mask on this data set. We'll be using some of them later. You do generally want to increase your significant digits up a little bit. And then we'll go ahead and show the intermediates and generate some of the contribution images so you can see what those look like. And we'll only fit one Gaussian to this. You can put in a directory to save your data to. So I'm going to go ahead and make a new directory, and you can just drag and drop that right in there. That will automatically save a lot of the data here. So if we get OK, that's going to go ahead and start the process. You do get a status bar with some status updates. Not all of these steps take an equal amount of time. Often the radial profile takes quite a bit longer. Any more questions? Doesn't look like it. I don't think I can see the QA section. I hope no one's been posting there. Some issues that you might run into. I tested this on 32 gigabytes, and most of these are relatively small, but CCC does use a fair amount of memory to do this, and these are the two biggest images in the example data set that we'll be using. So it may eat up a lot of the memory in order to do this. If you do get an out of memory error, if you uncheck the boxes to show the intermediate images and to generate the contribution images, that does reduce the memory usage. Additionally, in here, if you do the just cross correlation, you'll get the same options. I'm sorry, not my apologies. The CCC no confidence, triple C no confidence does effectively the same thing. You just won't get a confidence value with that data, and it does reduce the memory load. So once it's all done, we get a table of our results. So you can see that we have a mean value of 110 nanometers. By the way, I forgot to cover what these images are. So this is storm data out of that lab. So it's very good, clean storm data, 20 nanometers resolution. So we get a mean of about 118 nanometers, standard deviation of 184 nanometers. So very good result. Our confidence is very high at greater than 0.5. I will say that these results are even better when you use the full data set. So I took a pretty small cropping of this data in order to set up this tutorial. You do get even more precise results with a higher confidence with the full data. We get our contribution images, and you can feel free to look at those if you want. When you're doing this, and you can actually see it here, I can't adjust the brightness and contrast through the plugin, or at least I've not figured out a way to do that. So you will probably need to adjust the brightness and contrast. When you're looking at the contribution images, that usually will auto contrast just fine. But with these cross correlation result images, so these are, there should be 3 of these, one for the subtracted, one for the original, and then one for the Gaussian modified. These are the direct images that are used in the intermediate steps to calculate that correlogram. So here's our original cross correlation. If we subtract the low frequency data, we get this, right? And then after we calculate what that Gaussian curve fits to, we have this Gaussian modified. So this is what our correlation actually looks like in this image. So if you look at your original images, this is kind of the range of our correlation here, which looks pretty big, but just realize this is storm data, so it's very, very precise. Any questions about any of that so far? Not seeing anything. I will say mostly these cross-correlation results, they're not typically used. They can be fun to look at, but usually you don't need this information. The contribution images, sometimes people are using it, but again, it's not absolutely necessary. Mostly what you care about is right here, the correlogram. You can see that this Gaussian curve did not fit great, and I do think that's partially because I've cropped the image down so much. And I suspect that a lot of it has to do with this little dot right here, where we have higher cross correlation for one specific point. I'm not really sure what generated that, but that would mess up the algorithm, because in order to fit data near 0 like this, C actually mirrors this data from the peak. So it finds the peak of the data and then mirrors it across the other side, because the fitting algorithm that I have does not do well with bounded data. So I had found out that mirroring the data works pretty well. But when you have random peaks like that, it can cause issues. Does not happen too frequently. And you can see it still fit pretty close to where it should be. And especially if you have multiple images, for your experimental repeats, it's just not should be a major problem. Additionally, you are also given the full value for this table. So every single value that's used in this table, you get over here, and this will be automatically saved as well. This is not all the data. This is just so that you can actually see where your correlation is. If you want to see everything, you can go auto range both axes. And I probably shouldn't do that, because this is going to take a while. We have a pretty long distance here. So this goes 10 microns out, and that's where our data is. So this does search along the entire cross-correlation result image in order to find that correlation. Okay. There's no questions about that one. Oh, it's correlogram also comparing the colocalization with random distribution of the same proteins in the same area of volume. No, it's not. So you're talking about the Costes. It is not doing that. That is something I thought about implementing. That's kind of where the confidence effectively came from, was me trying to do a simplified version of that. The problem is that would use so much more memory to actually do, and takes significantly longer repeating this process over and over again in order to basically get a distribution. Also, the few times that I did test this, it wasn't really helpful. Much like Costa's significance test, when you do that, you tend to get different results. When you randomize things, it's going to be different. So I don't think it's actually terribly beneficial, and I think the confidence value is kind of the better metric here, where we're basically just directly measuring that low spatial frequency component in order to identify, do we have the resolution necessary to answer that question. So, all right. So we'll go ahead and move on to the next result here. Looks like I'm doing, sorry, next result, next example. If you are unable to analyze these for memory reasons, I did put all the results, at least the basics of them, into the folders here. Hopefully everybody's having success with this. The next result does have a mask, so you will need to drag and drop the mask. And in this, we have a bit of insufficient resolution. So you can actually pretty well see this. This came from a forum post, actually, someone asking for help with CCC. Would rotating one of the channels by 90 degrees be an appropriate internal negative control? I don't know. I've never really thought about that. Maybe. I really feel like the better solution is just to have multiple experimental repeats. If you're getting consistent mean, standard deviation, and especially height across multiple experimental repeats, I don't think you would necessarily need an internal negative control. Like I said, it's fairly different than pixel-wise, because we're not just comparing pixel by pixel. We're looking across the entire image. So what you really want to be looking for is like narrow standard deviations, high confidence, and then just results that make sense. So in this case, actually, this is supposed to be a fusion protein, so GFP bound to RFP directly. And it's supposed to be within the endoplasmic reticulum or mitochondria, I believe. I can't remember exactly. But either way, it does not look like that. And you can kind of tell just from looking at it that it is a little probably low resolution for the fluorophore that we have. But we'll go ahead and look at it by CCC anyway. And this data will look familiar. So we pick our two different images, one for each of those. This time we do have a mask, so we're going to uncheck that box and then make sure that we select our mask here. We go ahead and just delete our output directory, which I just realized I forgot to show you. And we're going to set our number of Gaussians to fit to 2. You'll see why in just a second. I'm also going to, well, yeah, we'll leave that check for now. And then click OK. This one should go quite a bit faster. It's a fair bit smaller. So, and there are our results. So we have our contribution images, and then we have our Gaussian modified results. So you can actually see the difference in that 2 fit there. So when we look at the parallelogram, this is where I got that data from. We have that really broad distribution with a small Gaussian on top. And you can see that in the Gaussian modified results. We have our basically close narrow band here along with the broad. And honestly, the reason I say that this is insufficient resolution, we do get that narrow band result, but this low frequency probably shouldn't be here. And that's why I say I think there's insufficient resolution with this data, because realistically, especially given that this is supposed to be within the mitochondria or endoplasmic reticulum, we shouldn't be getting that broad signal, especially with an appropriate mask. So just something to think about when you're analyzing your data. Make sure that your results obviously make sense. But with this, we do get the results for both of these Gaussian fits. So we have our broad one here, standard deviation of about 4 microns. That's basically we're correlating to the cell, the confines of the cell. And we're getting that because even though we're subtracting the background, we just have signal absolutely everywhere within the cell. And then we also get our narrow band, which is the 0.34, which is probably around the resolution limit of the microscope. So that's a pretty good result in that aspect. And if we cleaned up this data, maybe got a little bit more resolution, or figured out why it may not be localizing correctly, you could probably get a really good result with this in the end. So close all that. When you do check all the boxes, it generates a ton of data. Sorry about that. And just to show you on that last one too, we did get saved everything. If you don't have the contribution images or intermediate images checked, it will not save these, obviously. If you do have them checked, you will automatically save those as well. Okay. So the contribution image, sorry, I should have read that out loud. The contribution image is intersection and of two channels for visualization purposes only. And no, you don't have to use it just for visualization purposes. So actually, let's go ahead and take a look at these two contribution images. And they do come out as two separate stacks. Sorry about that. It's just a lot easier to program that way. But we can go ahead and merge these. And then I will change the channel here. So, oops, that was the wrong one. So yeah, the kind of are for visualization purposes. You can do analysis on these in terms of basically figuring out what particles analyze. But the contribution is basically what data falls within the Gaussian distribution across the other channel. So you can see in the original image here, if I open that up, that we have, close that for a second, some data kind of out here, and all of that is now missing from our contribution image. So we're only showing the data really that is correlating, right, within that spatial correlation range. And it looks like we have just kind of a couple there, which is why we have some out there. But that's the basic idea. And you can do some analysis on this. The one thing you will find is that the intensity values here are absolutely massive, right? And they correlate in no way to the intensity values in the original image. And that is by design. That's because I cannot figure out a normalization scheme to get it so that any of the values here reflect how much of this data actually contributed to that result. So rather than even trying, I make it so that it does give you just absolutely crazy values so that people don't think that there's any correlation in terms of the intensity here to here. The relative intensities within the image do have a meaning, right? So brighter intensities within the image mean that contributed more or less. Could also mean that the intensity in the original image was brighter. So the relative intensity can matter. It is very tricky to identify, but mostly the localization is what you may want to be analyzing here. Okay, does that answer your question? Awesome. Okay. Yeah, the contribution images, they're a little bit confusing, especially at first, but not too bad. So for our next example here, we're actually going to basically run through everything from start to finish. So like I said, we're going to be doing that mitosis sample. You can do this yourself using these instructions here. There are also a bunch of additional notes when you're doing this. I'm going to try and do this from memory and not mess up here. So the first thing we're going to do is go to File, Open Samples, and go to mitosis. I should say this is, in terms of correlation, like this is a terrible image for this. For demonstrating CCC, this is just the only time-lapse image that I have that's anywhere near decent. If anyone has time-lapse colocalization or cross-correlation data and wants to share it, I would love that. So this is the data we're working with. I already messed up because I didn't want to split the channels yet. One of the first things that we want to do, actually, because this is originally 16-bit data and it's easier to do this with just both channels here while they're open, is go to image, type 32-bit. And again, we're doing that just so that we can get negative values, right? If you leave it as 16-bit, you will not be able to have negative values. And then we are going to go to image, color, split channels. Now my memory is failing me because I don't remember which. We use channel 1 to generate the mask. Oh God, what did I do? There we go. So yeah, channel 1 here, we're going to right click, duplicate, create a mask. There are other ways to do that if you want. And then we're going to do a Gaussian blur. So process, filters, Gaussian blur 3D. Default values seem to work fine. I just did the wrong one. That's okay. Rename that, copy that, name that to mask. That to that. Hopefully you don't make that mistake. After we do that Gaussian blur, I should definitely leave this open because I don't remember all the values here. 2000. We do a threshold of 2000. And this is pretty low. I couldn't get any of the algorithms to get a decent mask here. I don't know why I'm trying to do that by hand. Type in 2000. Go. And you see this looks pretty broad, right? This is not a mask you would normally use, but what we're trying to get here is basically the area of the cell as a whole, right? Because that is where our proteins are confined to. Okay, so the mask generation with CCC is quite a bit different. A lot of these algorithms will not work necessarily very well. But with that, we'll go ahead and apply. When this comes up, because it's 32-bit, we'll hit convert to mask. We're going to hit uncheck calculate threshold for each image. And that is entirely up to you. I like the black background, so. Okay. Once we have that, got our mask. And now the next thing that we need to do is actually subtract the background from these images, both of them. And I'm going to show you a way I like to do it. And this works pretty well for most 3D data. It actually doesn't work great for these images. I just wanted to use it just so that you see. And if you go to the search bar here in Fiji, for those of you who aren't familiar, in the bottom right of the Fiji window, if you click there, there's a little search bar and you type in median, down in the ops section, make sure that the ops box is checked. There is this stats.median, and this can give you the median value for an entire image. You hit run on that, and then you select your image down here. So we'll do channel 1 first and hit OK. And that gives us our median value. So this is the median of all the pixel intensity across the entire image. Now here we do have a lot of data that's kind of within the cell, but not necessarily in the background, which is back here. This probably isn't the best method for this. It would probably be better to measure the background, but this is a lot faster and easier. And again, we're not doing this for real purposes. And I will say for most 3D data, this method will work because most of the pixels in the image are background. Anyway, we will go process, math, subtract. Actually, I'm going to make sure I selected that image last. I don't want to mess this up. Math, subtract, 2076. Click OK. Yes. And there is our background subtracted, right? And so that is just getting the background here to around 0, which is what we want, okay? If you want to play around with it, you can not do that and see the difference. The confidence values will be extraordinarily low. And then we're just going to repeat the same thing with channel 2 here. So median, stats.median, select channel 2, click OK, 2629. So come here, process, math, subtract, 2629. Click OK. Yes. So there we go. There are 2 images. Okay, and these are what we're going to use. So now we will go to Analyze, Colocalization, run CCC again. You can also search for that if you just show you really quickly. If you just search CCC, it'll pop right up. Hit run. So image 1, channel 2, and then make sure you have the mask selected. All of that will be the same, and then hit OK. And I accidentally left Gaussian set to 2, which doesn't work great with this data, but that's okay. And this does take a little while. While this is going, I should explain what's going on. So it is analyzing each frame independently, right? And so this doesn't actually use as much memory as you might think, because we're not cross-correlating both of these images all together at the same time. We're going frame by frame, taking just that frame, analyzing the cross-correlation, fitting a Gaussian curve to that data, and then moving on to the next frame, and also concatenating the contribution images, concatenating together the intermediate images. And so all the data that you get is on a frame by frame basis, and you'll see that in just a second here. So the output does look a little bit different. So here we have our table of results. So this is not the table of the original cross-correlation data. These are our Gaussian fits and confidence values and R squared and everything. And this is for every single time point. So you get a table of all your data right here. It also goes through and finds where it had the highest confidence. Looks like right here. And gives you that as kind of a summary of where it found the highest confidence value, what the mean was, the standard deviation was, etc., just for that one single frame. Okay. And it reports the time that it gives. If you need to find out what frame that is, you can just go to the correlation over time. 3.286 is frame 24. So again, the contribution images are calculated per frame. So every single frame gets the contribution using the Gaussian curve fit from that frame to generate that image, which means the contribution images, the value of the Gaussian changes over time, just like it does in the results. Definitely a little confusing. If you want to basically see the contribution for a set value, if you just want to see, what data correlates with a mean distance of 1.5 and a standard deviation of 1, you can do that. You just have to go to Analyze, Colocalization, and do Custom Contribution Images. When you do that, you can actually generate your own contribution. And with time-lapse data, you can set it so you can just say, I want to see all the correlating particles that are at this specific range. And that could be useful if, say, you're expecting over time two particles to come together, and you basically just want to visualize which particles are localized together in your data or at that spatial correlation within your data over time. One other unique thing here is this is the correlogram that we have. I need to rename this. This is our correlogram. It's actually an image, and originally it's going to be really hard to read. And this is because this axis here, our y-axis, is the distance axis, which can be absolutely huge, as I already showed you. So the 51 here is our time axis. So we're looking at time over distance. So the way to use this is obviously to crop it down. So what we're going to do, select that image, go edit selection. Oh, wow, I only have 8 minutes left. Edit selection, specify. Make sure the width is 51. Height is, we'll do about 1500, and then both of those should be 0. Click OK, and then right click in there, duplicate. And so this is a heat map of our correlogram for every single time point. Okay. All right. And since I am running low on time, I'm going to go back to the PowerPoint here because there's a little bit more that I wanted to go over. Were there any more questions? Doesn't look like it. If there are, I can stay a little late afterwards, but I just wanted to mention a few things. So, oops, I am now sharing the wrong screen here. 1 second. Just do that. Looks like I'm still sharing. All right. So now that you've seen how everything works, I just wanted to mention that CCC is scripting and PyImageJ compatible. So if you're using PyImageJ, you can initialize it with colocalization by cross-correlation. And then this is an example Groovy script right here with all of the parameters showing. Many of these are optional. You don't have to use them. And then I also have an example on the GitHub for colocalization by cross-correlation example script where it concatenates the results across multiple different analyses into one big table. Few recommendations for reporting this data on CCZ. I do not recommend reporting it in a table like I did in my publication. That was probably a bad move. I did that mostly because I was trying to show very specifically which parameters changed with very controlled conditions if you ever go to look at the publication for CCC. And on that note, please reference me if you use the plugin. What I would recommend is doing something like this, right? So creating a graph of the Gaussians that are made from the average parameters for each condition. I will get to that question in just 1 second here. That's fine. Let me finish this up. So yeah, a graph of the Gaussian of the average parameters. And I made this with something called Desmos. This is a free online tool. There is a link here that goes to a calculator just for this. But it's basically the equation for a Gaussian curve, not a normal distribution, which is definitely different, especially if you talk to stats people, make sure you emphasize that it's not a normal distribution. But the equation for a Gaussian curve twice over, so that you can compare them side by side. Now, obviously, you can't see the variation when you do this. If you want to see the variation and to do stats on this, what you want to do is actually make a scatter plot of the three parameters for the Gaussian curve, the height, the mean, and standard deviation. You don't have to do all three. There are definitely reasons you may want to omit the height, such as you do not have equal intensity across the images. So if you have your green channel is a transfected channel, those can vary a lot in intensity. So you may want to omit the height in that case, just because that can strongly affect the height. But anyway, this is a good way to basically visualize the variance within your data, within each channel. And then to do stats on multiple of these, you can do a Hotelling's multivariate two-sample t-squared test for significance. This can take 2 or 3 of these parameters in order to do statistics on this. Don't use the confidence for stats. It's not what it's there for. Like I said, it's there for filtering and results. This has already been done by someone. It's too late. It's already been published, so I can't really do anything about it. But please don't do that. Also on that note, all the cross-correlation values in the correlogram and in the reported values and the Gaussian height are all normalized to the mass volume. And this was done so that if you accidentally have, cells that are twice the size, or if you accidentally have 2 cells that are touching when you put the mask in, or just doing it across an entire image with multiple cells, you will get normalized data to that mask volume. And then last note on this, please talk to a statistician. I am not a statistician. I did talk to a statistician about some of this, but this is not my area of expertise, and your data may be different. CCC is also very new. This has not been done a lot, so definitely good to go and talk to someone about this. Okay, let me take a break really quickly and answer Estefan's question here. How is the correlogram affected when analyzing elongated structures like fibers instead of punch or globular structures? Yeah, so you will get a slight broadening of the error measure, the standard deviation, right? A little bit. It's actually not affected too much, and you can actually see that. I'm not going to go back. Puncta, yeah, makes sense. I'm not going to go back right now, but if you look at the endoplasmic reticulum versus mitochondria, right, that is that case. And a lot of the kind of spreading out there of that standard deviation was more just because of the abundance of the endoplasmic reticulum and the mitochondria, not so much because they're elongated structures. But this is actually one of the things that CCC excels at, is those type of structures, because there's not really a great other way to analyze elongated structures. Like you can't do distance analysis on it very well, especially if both structures are elongated, because all the distance analysis algorithms usually use a centroid. So if your object does not have a clear, well-defined center, it kind of ruins that. So CCC actually works really, really well with that data. Again, it broadens that distribution a little bit, but you still should get a good mean value. So, okay. All right, some troubleshooting recommendations. If you're having problems with CCC, I highly, highly recommend that you use the autosave feature and that you look at the correlograms. This is one of the first places I always look when people are having problems. If you're getting weird results, all the parameters summarize the data that you see in the correlogram. So looking at the correlogram, you're going to get a much, much clearer picture. As I already mentioned, you can actually see and visualize the z-step in 3D data. So when you have 3D data, you will usually see the z-step in here. There are some common characteristics with the z-steps. You'll often see drops in the z-steps, and this can indicate sample anisotropy. So basically, you have cells growing flat on a cover slip. They grow out and not up very much, so they tend to have features that are more lateral than axial. And that tends to correlate with a drop in cross-correlation. You can also get increases. Like you can see here, cross-correlation is going down, and then we get a jump with the z-step. And this is often due to PSF anisotropy, which is totally normal. But if you have really large jumps, or if this seems like it's just not going down fast enough, that can indicate spherical aberration. Some other things to look for: if you have really low height or a very, very narrow standard deviation, especially below the resolution limit, that can mean that you have a really dim channel, no true correlation, or just insufficient data. Basically, your image is too small. If you get negative values, typically means abstract the background too much. And then if you do not get a fit at all, if CCC just cannot find any data, you'll just get this broad flat distribution here. You also get negative confidence with that. Okay, moving on. Summarizing here, trying to finish up really quickly. I know we're already at 1 o'clock. Sorry about that. There are a lot of pros and some cons with CCC. So we do get results improved with our resolution, and it's compatible with super resolution data. It's 3D and time compatible, which is nice. It is also headless mode compatible. You should be able to do this on a cluster. I've never tested that myself. I don't have access to one, but it should be compatible with that. And like I said, it's scripting compatible, both Groovy and PyImageJ. This does not work great in ImageJ macro because the ImageJ cannot read from the SciJava tables. Anyway, it's also relatively easy to execute, especially in comparison to other cross-correlation algorithms. Some of the cons: it does use a lot of memory. The images that we were doing today were particularly small. Once you get big images, you're going to notice it requires a lot of computer memory. It's also very sensitive to mask and the background. And the results can be confusing. There's 5 output parameters, and the statistical analysis is relatively involved. So there may be future improvements coming soon. Hopefully there will be a memory improvement soon, so it uses a little bit less memory, or at least doesn't require as much. And then last thing here, sorry for going over a little bit, but I do want to acknowledge some people. So first, the organizers, thank you very much for having me. I also wanted to say thank you to the University of Buffalo School of Dental Medicine for supporting me through this. And then lastly, 2 teams that were just, I could not have done this without these libraries existing. The ImageLib2 team, the FFT convolution class, which actually does the cross correlation, was not me. That was the ImageLib2 team. So CCC would not exist without them. And then same with SciJava and Fiji team. SciJava made this so much easier. I didn't have to design a single UI thing at all. So with that, if there's any questions, you can definitely stick around now or post on the forum. And obviously this image has nothing to do with CCC. I just like the animation. So